Regulation of gene expression. Importance of gene regulation. Regulation of gene expression or gene regulation is essential for prokaryotes and eukaryotes. As it increases the versatility and adaptability of an organism by allowing the cell to express protein when needed. Furthermore, gene regulation drives the process of cellular differentiation and morphogenesis, leading to creation of different cell types in multicellular organisms, where the different types of cells may possess different gene expression profiles, though they all possess the same genome sequence. Methods of gene regulation. There are two possible ways of regulation of gene expression: positive regulation and negative regulation. When the expression of gene is quantitatively increased by the presence of specific regulatory protein, the activator is called positive gene regulation. Whereas when expression of gene is diminished by the presence of specific regulatory protein, the repressor is called negative gene regulation. Lack of dual positive and negative control of gene expression. A classic example of positive regulation in bacteria is lack of responsible for obtaining energy from beta galactosides such as lactose. In bacteria, genes are clustered into ophirons, which are genes clustered that encode the proteins necessary to perform coordinated functions such as catabolism of a substrate obtained from outside like ophron or biosynthesis of a given amino acid. RNA that is transcribed from prokaryotic ophirons is polycistronic, a term implying that multiple proteins are encoded in single transcript. The leg ophron consists of one regulatory gene and three structural genes Z, Y, and A. The I gene codes for repressor of leg ophron. The Z gene codes for beta galactosidase, which is primarily responsible for the hydrolysis of disaccharide lactose into its monomeric units, galactose and glucose. The Y gene codes for permease, which increases permeability of cell to beta galactosides. The A gene encodes a transacetylase, an enzyme that transfers an acetyl group from acetyl coenzyme A to beta galactosides. Its precise function as part of leg ophirone is not understood currently. Mutation. A gene mutation is a permanent change in DNA sequence that makes up a new allele in the population. Mutations range in size from change in a single DNA nucleotide to a large segment of a chromosome or whole chromosome, or sometimes change in number of chromosomes. Mutations are caused by radiation, viruses, transposons, and mutagenic chemical, as well as errors that occur during meiosis or DNA replication. These agents that cause mutations are called mutagens. while the organism in which mutation is occurred is called mutant. Some mutations are very rare, others are common in population. Mutations that occur in more than 1% of population are called polymorphisms. They are common enough to be considered a normal variation in the DNA. Polymorphisms are responsible for many of the normal differences between people, such as eye color, hair color, and blood type. Although many polymorphisms have no negative effects on a person's health, some of these variations may influence the risk of developing certain disorders. Origin of mutation. Gene mutation occur in two ways. They can be inherited from a parent or acquired during a person's lifetime. Mutations that are passed from parent to child are called hereditary mutations or germline mutations because they are present in the egg and sperm cells, which are also called germ cells. This type of mutation is present throughout a person's life in virtually every cell in the body. Mutations that occur only in an egg or sperm cell are those that occur just after fertilization are called de novo mutations. The no mutation may explain genetic disorders in which an affected child has mutation in every cell but has no family history of the disorder. Acquired or somatic mutations occur in the DNA of individual cells. At some time during a person's life, these changes can be caused by environmental factors such as ultraviolet radiation from the sun, or can occur if a mistake is made as DNA copies itself during cell division. Acquired mutations in somatic cells other than sperm and egg cells cannot be passed on to the next generation. Types of mutation. Mutations are of two types on the basis of how mutation occurs in the cells. The mutation which occurs naturally and automatically due to internal factors are called spontaneous mutations, whereas the mutations which are produced by external factors are called induced mutations. Mutations are of two types on the basis of where the mutation occurs and to what extent. A mutation that causes change of single or few nucleotides in the DNA is called point mutation, whereas the mutation that causes change in structure of number of chromosomes is called chromosomal mutation or chromosomal aberration. Point mutation involves a sudden change in the sequence of nucleotides of a gene that causes change in the phenotype of an organism. Such mutations occur in following ways. It is the removal of one or few nucleotides from a particular segment of DNA. It is the addition of one or few nucleotides in a particular segment of DNA. For example, this is inversion. Deletion, it is the removal of one or few nucleotides from a particular segment of DNA. Inversion, it is the addition of one or few nucleotides in a particular segment of DNA. This is the example of deletion. 1T is removed. This is the example of inversion. TT replaced by double A. Base substitution, it is replacement of one of few nucleotides in a particular segment of DNA. C replaced by A. Structural changes in chromosome are type of chromosomal mutation or aberration. Such changes in chromosomes take place during meiosis. When due to certain mutagens, chromosome is split down into several fragments, but later on, when these fragments reunite, its new pattern becomes changed from original one. Structural changes in chromosomes are of following type. Deletion, it is the removal of a segment of chromosome comprising single or few genes. For example, this is the example of deletion. You know, inversion. In this case, a portion of chromosome breaks up, turns around and joins again in such a way that the sequence of gene gets reversed. This is inversion. Translocation. It involves shifting of a segment of one chromosome to another. Non-homologous chromosome, thus both the chromosomes are affected. The donor chromosome suffers a deletion while the recipient chromosome becomes longer than normal. For example, this is the example, Duplication. It is the repetition of one or few genes and same chromosome. For example, this is the example of duplication. Another aspect, chromosomal aberration, is the change in number of chromosomes, which occurs due to chromosomal non-disjunction during meiosis. Change in the number of chromosomes due to addition or loss of one or more chromosome is called aneuploidy, whereas the change in number of chromosomes due to addition or loss of one or more complete set of chromosomes is called euploidy. Aneuploidy may be of the following type: monosomy, 2n minus 1. It is the result of loss of single chromosome from the diploid set. Nullisomy, 2n minus 2. It is the result of loss of a pair of homologous chromosome from the diploid set. Trisomy, 2n plus 1. It is the result of addition of single chromosome in the diploid set, for example, Down syndrome and Klinefelter syndrome. Tetrasomy, 2n plus 2. It is the result of addition of pair of homologous chromosomes in the diploid set. Euploidy is the state of a cell or organism having an integral multiple of the monoploid single set number of chromosome. It is called polyploidy. It exists in various forms like triploidy, 3 sets, tetraploidy, 4 sets, pentaploidy, 5 sets, hexaploidy, 6 sets, and so on. As a general rule, polyploids can be tolerated in plants but are rarely found in animals. This is most important point. One reason is that the sex balance is important in animals and variation from the diploid number result in sterility. Types of mutagens. Mutagens can be artificially produced, induced mutation by certain agents called mutagens or mutagenic agents. Following are two major types of mutagens. Physical mutagens. Short wave radiation are the most important physical mutagens. H. J. Muller was the first to induce mutation using X-rays and Drosophila. Other than X-rays, gamma rays and ultraviolet radiation can be used to induce mutations. The main source of spontaneous mutation is the natural radiation coming from cosmic rays of the sun. They occur in small amount in environment and are known as background radiations. Radiation cause breaks in the chromosome. These cells then show abnormal cell divisions. Different types of cancers are the result of radiation. Ultraviolet rays affect the structure of DNA helix and also affect the replication process. Chemical mutagens. A number of chemical aids as mutagens such as nitrous acid, formaldehyde, mustard gas, fibromo, acidins, etc. Chemicals such as colchicine induce polyploidy in cells. Caffeine, nicotine, food preservatives, and pesticides are also mutagenic. Remember this point. These are very dangerous for mutation and human health. The first chemical mutagen discovered was mustard gas that was used as chemical weapon during World War I. Diseases induced by mutation. Sickle cell anemia. Sickle cell anemia is an autosomal recessive genetic blood disorder characterized by abnormal rigid sickle-shaped red blood cells. Sickle shape decreases the cell flexibility due to which they can also get stuck more easily in small blood vessels and breaks into pieces that interrupt healthy blood flow. Sign and symptoms.